You know, you could have the strongest, most resilient heart on earth. You could have these flawless lungs capable of pulling in, I don't know, gallons of pristine air with every breath.
Right?
But without one specific, highly sensitive, shape-shifting protein patrolling your bloodstream, you would literally suffocate in a matter of seconds.
Yeah, it's a really sobering thought, isn't it? I mean, we focus so much on the macroscopic organs like the pumps and the bellows, right?
Exactly, the big stuff.
Right, the big stuff. And we just completely overlook the molecular machinery that's actually doing all the heavy lifting.
And that machinery is exactly what we're focusing on in today's deep dive. We are taking a stack of sources spanning, gosh, 150 years of physiological history, all the way to modern medical trials.
Oh, yeah, some really fascinating recent trials.
Right. And our mission is to understand the intricate chemical dance of how your blood actually transports oxygen and carbon dioxide.
Which, honestly, is a process that is fundamentally misunderstood by almost everyone.
Yeah, let's talk about that right out of the gate.
So the standard assumption, and this was actually shared by the most prominent physiologists of the mid-1800s, by the way, is that gas transport in the blood is just, well, a matter of physical solubility.
Like you breathe oxygen in and it just kind of dissolves into the liquid plasma?
Exactly. Much like carbon dioxide dissolving into water to create, a carbonated soda.
Which is super intuitive, right? I mean, the lungs expose the blood to a gas, the liquid absorbs it, and off it goes to your brain and your muscles.
It is intuitive.
Yeah.
But it's mathematically impossible.
Wait, really? Impossible?
Totally impossible.
Yeah.
When 19th century researchers finally sat down and calculated the physical solubility of oxygen in plasma, that whole soda water theory just completely collapsed.
Huh.
Because at normal human body temperature and at the typical oxygen pressure found in your arterial blood, plasma can only passively dissolve about 0.3 milliliters of oxygen per 100 milliliters of blood.
Okay, let me put that in perspective for you. Because a standard resting adult needs about 250 milliliters of oxygen per minute just to maintain baseline cellular function.
Right, just sitting there on the couch.
Exactly. So if we were relying purely on dissolved oxygen in the plasma, our circulatory system would fail to deliver even like 2% of what we need to survive.
Yeah, you would essentially be asking a tiny trickle of water to put out a roaring forest fire.
Wow.
The transportation infrastructure of just the blood plasma alone is totally inadequate for human metabolic demands. So evolution had this massive solubility bottleneck to overcome.
The solution couldn't just be passive, right?
Right. It couldn't just be a passive physical process. It had to be a highly active chemical workaround.
Okay, let's unpack this. Because trying to transport oxygen purely by dissolving it in plasma is, well, it's like trying to evacuate a massive stadium using only single-seater bicycles.
I like that.
You need a bullet train, right? And that chemical workaround, that bullet train, is hemoglobin.
Exactly. And it's a huge shift in thinking. Instead of relying on physics, just letting gas float in a liquid, the body relies on biochemistry to actively grab, bind, and carry the payload.
Who was it that figured this out?
That was Felix Hopseiler. Back in 1862, he isolated the oxygen-carrying pigment of blood, crystallized it, and he named it hemoglobin.
1862, wow.
Yeah, and crucially, he demonstrated that this wasn't just a passive suspension. Hemoglobin forms a reversible chemical bond with oxygen. It's an active carrier.
And the sheer capacity of this carrier is what keeps us all alive, because looking at the sources, in 1894, a guy named Gustav von Höfner calculated the exact binding capacity.
Oh yes, Höfner's number.
Right, Höfner's number. And he found that 1 single gram of hemoglobin can bind roughly 1.34 to 1.36 milliliters of oxygen.
And since a healthy adult has about, roughly 15 grams of hemoglobin per 100 milliliters of blood, the math completely transforms.
Right.
Instead of carrying that measly 0.3 milliliters of oxygen, your blood carries about 20 milliliters per 100 milliliters.
Which is nearly a 70-fold increase in carrying capacity. It completely bypasses the physical limits of solubility.
That's incredible.
And, this isn't just dry textbook science for you to memorize. This exact bottleneck is why anemia causes such crushing fatigue. Absolutely. Because if your hemoglobin count drops, it doesn't matter how robust your heart or your lungs are. The chemical carriers are missing. The bullet train is missing cars, basically.
That's the clinical reality of it, yeah. The gas exchange in your lungs might be absolutely perfect, but without enough hemoglobin to pull that oxygen out of the solution and ferry it away, the whole system just stalls out.
Okay, but wait. This introduces a pretty massive logistical flaw in my mind.
Oh, What's that?
If hemoglobin has such an intense affinity for oxygen, like if it's such a powerful chemical magnet that it strips oxygen out of the lungs, how does it know when to let go?
That is the million-dollar question.
Right. High affinity is great for loading up the bullet train, but it seems like a disaster for unloading. Wouldn't it just hoard the oxygen all the way through the body?
And that is the exact paradox that early 20th century physiologists wrestled with.
Right.
Because hemoglobin couldn't just be a static bucket. It required some sort of mechanism to differentiate between the lungs, where it needs to bind oxygen tightly, and the deep tissues, where it desperately needs to release it.
It had to act as like a localized sensor.
Precisely. And what's fascinating here is that in 1904, the Danish physiologist Christian Bohr discovered exactly how it works.
The Bohr effect.
Yes. Bohr observed that hemoglobin's affinity for oxygen isn't a fixed constant. It's highly sensitive to its immediate chemical environment.
So it changes depending on where it is.
Exactly. Specifically, he found that when carbon dioxide levels rise and the pH of the blood drops, meaning it becomes slightly more acidic, hemoglobin is chemically forced to weaken its grip on the oxygen.
Which is remarkably elegant because think about it, where do we find high carbon dioxide and low pH?
In active metabolizing tissues.
Yes. The exhaust products of cellular respiration actively force the hemoglobin to drop its payload right where the fuel is needed most.
And the numbers track this beautifully, by the way. In the lungs, where oxygen is plentiful and carbon dioxide is very low, hemoglobin is about 97 to 98% saturated. It's fully loaded.
Right, packed full.
But then, when it reaches resting tissue, the local CO2 causes it to release some of that oxygen, dropping its saturation to about 75%.
So it leaves about a quarter of its payload just for basic, resting maintenance.
Yes. But say you suddenly start sprinting. Your leg muscles are going to generate a massive surge of carbon dioxide and acidic metabolic byproducts.
Lactic acid, that sort of thing.
Exactly. And that severe drop in local pH triggers a strong Bohr effect. It shifts the hemoglobin binding curve sharply to the right.
So what does that actually mean for the oxygen?
It means that in that highly acidic environment, hemoglobin might dump 40 to 50% of its oxygen locally. It literally responds to the metabolic demand of the tissue in real time.
So it's delivering fuel based entirely on the presence of the exhaust. That is wild.
It really is.
But, how is a single protein physically capable of doing that? Proteins don't have brains, right? They can't sense anything in a cognitive way.
No, not at all.
So what is the actual atomic mechanism that makes this happen?
Well, to understand that, we have to fast forward a bit to 1962. That's when Max Perutz won the Nobel Prize for mapping hemoglobin's atomic structure. He used X-ray crystallography.
Okay.
And he found that hemoglobin isn't just a single solid block. It's actually composed of 4 separate subunits.
4 individual pieces.
Right. And Perutz discovered that the entire 4-part structure physically flexes and contorts between 2 different shapes, depending on what it's binding to.
Wait, it physically changes shape?
Yes. He termed them the tense state, or T state, and the relaxed state, or R state.
Oh, this is what we call cooperativity, right? I've always found the mechanics of this so fascinating.
It's one of the coolest concepts in biochemistry.
Okay, the analogy that always works for me here is an old, stubborn metal folding chair.
Okay, I like where this is going.
So you pull this rusty chair out of storage, and it's locked up tight. You yank on it, but the structural tension holds it completely shut.
Right.
That is hemoglobin in the T state, the tense state.
Wow.
It has a very low affinity for oxygen. It aggressively resists binding that first molecule.
It does, yeah. Because the physical bonds holding those subunits in the T state are quite strong.
Right. But if you apply enough pressure to force just one hinge on that chair to pop open, suddenly the structural tension of the whole frame changes. The other hinges loosen up, and the chair just snaps open effortlessly.
That is a brilliant way to picture it. In hemoglobin, once that first oxygen molecule finally forces its way into one of the subunits, it breaks these tiny connections called salt bridges.
Okay.
And the physical shape of the molecule shifts slightly toward the R state, the relaxed state. And that shift makes it exponentially easier for the second, third, and fourth oxygen molecules to bind.
It's like a profound structural communication across the protein.
Exactly.
Yeah.
But, the exact kinetics of how that folding chair snaps open, how that communication happens, that actually generated a massive debate in the 1960s.
Oh, really?
Yeah, it really gets to the heart of how molecular machines function.
You're talking about the debate between the MWC and TANF models, right?
Yes, the MWC versus KNF models. So the MWC model, which is also called the concerted model, argued that symmetry is always preserved.
Meaning what exactly?
Meaning the entire hemoglobin molecule flips from the T state to the R state all at once. The whole chair snaps open simultaneously.
Ah, I see. So if one subunit changes, all the other three have to instantly change to match it.
Exactly. But then there's the KNF model, the sequential model. And they argued it's more like a row of dominoes.
Okay.
One subunit binds oxygen, changes its shape, and that slightly nudges the neighboring subunit, which then nudges the next.
So it's progressive, not instant. Which one is right?
Well, textbooks often lean heavily on the MWC concerted model because it elegantly explains the mathematics of the binding curve, but the reality is much more nuanced.
It usually is.
Right. Modern spectroscopy shows that hemoglobin actually exhibits behaviors of both models. It's this incredibly complex allosteric machine where distant parts of the protein communicate through these tiny, tiny shifts in atomic geometry.
Man, that is amazing. Okay, so we have this intricate shape-shifting delivery mechanism dropping off oxygen specifically where the acidic carbon dioxide exhaust tells it to.
Right.
But that exhaust itself creates the next physiological crisis for the body. You can't just leave carbon dioxide building up in the tissues, or the pH drops too far and your cells just begin to die.
No, you absolutely have to clear it out.
Right.
And nature handles this with perfect reciprocal efficiency. The delivery of oxygen and the clearance of carbon dioxide are fundamentally the exact same mechanistic loop. And we know this thanks to John Scott Haldane.
Right, Haldane. He spent a significant portion of his career studying respiratory physiology and, interestingly, mine safety.
Yeah, quite the combo.
So while Bohr discovered that CO2 forces oxygen off hemoglobin, Haldane documented the reverse.
The Haldane effect.
Yes. Deoxygenated hemoglobin has a much higher affinity for carbon dioxide and hydrogen ions than oxygenated hemoglobin does.
Right. The physical act of the oxygen leaving the binding site actually reshapes the protein to better accept the acidic exhaust. They literally facilitate each other.
But let's clarify how that exhaust is actually traveling, because here's where it gets really interesting, and there is a massive misconception here.
Oh, definitely.
Most people assume carbon dioxide just dissolves into the blood as a gas and bubbles its way back to the lungs. But according to the sources, only about 7 to 10% of carbon dioxide is transported as a dissolved gas.
It's a very small fraction. Another 20 to 23% or so binds directly to the amino acids on the hemoglobin protein itself. That forms something called carbaminohemoglobin.
Okay, but wait. If you add those up, that still leaves about 70% of the carbon dioxide unaccounted for. Where does it go?
And this is where the biochemistry gets incredibly aggressive. That remaining 70% doesn't travel as CO2 at all.
Wait, what is it then?
It is instantly converted into bicarbonate ions inside the red blood cell.
Instantly. By what?
It's handled by an enzyme called carbonic anhydrase, which was discovered in the 1930s by Nona Meldrum and Francis Roughton.
Ah, right. And what stands out to me in the research is the sheer velocity of this enzyme. The sources note it catalyzes roughly 1 million reactions per second.
It's mind-boggling.
Honestly, how does a biological molecule operate at that speed without violating the physical limits of diffusion?
Well, it achieves that speed through brute force atomic manipulation.
Brute force.
Yeah. Carbonic anhydrase contains this highly coordinated zinc ion at its active site. And that zinc ion is so positively charged that it pulls on the electrons of a nearby water molecule, essentially splitting it.
Wow, okay.
This creates a highly reactive hydroxide ion. So when a carbon dioxide molecule drifts in, that hydroxide ion attacks it instantaneously, converting it into bicarbonate.
So it's basically setting a chemical trap.
Exactly. It is one of the fastest known enzymes in human biology.
I mean, it has to be, right? Because a red blood cell only spends about 3/4 of a second traversing a tissue capillary.
Right. You don't have time to waste.
If the enzyme operated at standard chemical rates, the blood would rush past before the CO2 could even be neutralized.
Yep.
But wait, converting all that CO2 into bicarbonate, doesn't that create an electrical problem?
Right.
You're generating massive amounts of negatively charged bicarbonate ions inside this tiny cellular envelope.
You are. And if that bicarbonate just sat inside the red blood cell, the chemical reaction would actually reach equilibrium and just halt entirely.
Which would be bad.
Very bad. So the cell pumps the bicarbonate out into the blood plasma.
Okay, but if you rapidly pump out negative charges, the inside of the cell becomes overwhelmingly positive, right? Which would destroy the whole electrochemical gradient of the cell.
Exactly. Which brings us to a really elegant solution. The hamburger shift.
The hamburger shift, also known as the chloride shift, discovered in 1892.
Yes. To maintain perfect electrical neutrality, The red blood cell has a specialized transport protein. For every negatively charged bicarbonate ion it ejects into the plasma, it pulls one negatively charged chloride ion inside.
It just swaps them.
It swaps them. The net electrical charge remains perfectly balanced, which allows carbonic anhydrase to just keep furiously churning out bicarbonate without ever stalling.
That is brilliant. So for you listening, 70% of the carbon dioxide you're exhaling right this second spent its journey up to your lungs disguised as a bicarbonate ion.
Yeah, and this reveals a staggering biological synthesis, really. If we connect this to the bigger picture, that bicarbonate floating in your plasma isn't just a clever transport disguise.
No.
It is the primary chemical buffer that prevents your blood from becoming dangerously acidic.
Oh, wow.
Your kidneys are constantly regulating systemic bicarbonate to maintain your acid-base balance. So the exhaust mechanism for cellular respiration is biochemically identical to the regulatory system that manages your blood's pH.
The architecture is just flawless. But, we can truly appreciate the precision of this system by examining what happens when it gets derailed, right?
Oh, for sure. When things go wrong, they go very wrong.
Right. So let's look at carbon monoxide. We all know it's lethal. Everyone has a detector in their house. But structurally, why does it shut the body down so completely?
Well, carbon monoxide poisoning is uniquely dangerous because it exploits the exact allosteric properties, that shape-shifting cooperativity we talked about, that make hemoglobin work in the first place.
It uses the system against us.
Exactly. It is a dual-pronged attack, a double whammy. First, carbon monoxide binds to the iron at the center of the hemoglobin subunit with roughly 200 to 250 times the affinity of oxygen.
So it just aggressively outcompetes oxygen for the physical seats on the bullet train?
It steals the seats, yes.
Yeah.
But if that were the only issue, it would just be like severe anemia. You just have less carrying capacity.
Which is bad, but maybe not instantly lethal.
Right. The real lethality comes from the second effect. When carbon monoxide binds to even one or two of the four subunits, it forces the entire hemoglobin molecule to lock into that relaxed, high-affinity R state.
Wait, meaning it violently shifts the dissociation curve to the left?
Exactly.
Yeah.
The hemoglobin holds onto whatever oxygen it does have with an absolute iron grip.
Oh, I see.
Even when it reaches the oxygen-starved tissues, the hemoglobin refuses to release its payload. Carbon monoxide doesn't just steal space, it turns your remaining oxygen into captive molecules that your tissues simply cannot access.
A complete allosteric trap. You have oxygen, but you're suffocating anyway.
Yes, exactly.
But, biology also uses this leftward shift, this high affinity state, to its advantage in specific healthy scenarios too, like fetal development, for instance. Surprising fact number 2.
Yes, fetal hemoglobin. It's basically a necessary biological heist.
The heist, I love that.
Well, think about it. The developing fetus cannot breathe air, right? It relies entirely on the mother's blood supply via the placenta. If the fetus and the mother had identical hemoglobin, the oxygen would simply equilibrate between them, and the fetus would suffer from hypoxia. It wouldn't get enough.
The fetus needs a chemical advantage to literally strip the oxygen away from the maternal supply.
And it achieves this through fetal hemoglobin, or HbF.
Yeah.
So adult hemoglobin is made of 2 alpha and 2 beta protein chains.
Mm-hmm.
Fetal hemoglobin replaces the beta chains with gamma chains.
What does that structural change actually do?
That subtle change means fetal hemoglobin doesn't bind as strongly to a molecule called 2-conol-3-BPG.
And 2-conol-3-BPG is the molecule that normally forces adult hemoglobin to release its oxygen, right?
Exactly. So because it ignores that release signal, fetal hemoglobin is naturally shifted to the left. It has a permanently higher affinity for oxygen than the mother's blood.
Wow.
So as the maternal blood passes through the placenta, the fetal hemoglobin actively rips the oxygen right off the adult carriers.
It's a beautifully engineered one-way ratchet system.
It really is.
But, knowing how tightly engineered this all is, it makes you wonder about modern medicine.
How so?
Well, given all the logistical nightmares of blood typing and donor shortages, why can't we just manufacture this stuff? Why have we failed to just synthesize vats of hemoglobin for trauma centers?
Well, it's not for lack of trying. The biotech industry poured billions into that exact idea in the 1990s and 2000s.
Okay.
They developed what are called hemoglobin-based oxygen carriers, or HBOCs, things like Baxter's Hemosys.
Hemosys, straight.
The logic was straightforward. You extract pure hemoglobin, strip away the red blood cell, and infuse the pure protein directly into trauma patients to restore oxygen delivery instantly.
Seems like it should work, but the clinical trials were catastrophic, right?
Right? They had to be completely aborted. Mortality rates actually spiked.
Why? What went wrong?
The researchers learned a very brutal lesson about physiological packaging. See, free hemoglobin roaming in the blood plasma aggressively scavenges nitric oxide from the endothelial cells that line your blood vessels.
And nitric oxide is crucial for keeping blood vessels relaxed and dilated.
Exactly. So when the free hemoglobin strips away the nitric oxide, the blood vessels violently constrict,
Oh, wow.
resulting in severe hypertension, massively reduced blood flow, and widespread tissue damage.
So the red blood cell isn't just a convenient little bag for carrying hemoglobin.
Not at all.
Yeah.
It is a vital biological quarantine. It prevents the highly reactive hemoglobin from basically tearing apart the surrounding cardiovascular infrastructure.
You cannot extract one gear from the evolutionary machine and expect it to function in isolation.
No, you really can't. The cellular packaging is just as vital as the protein inside.
Which brings us to a lingering mystery that the sources highlight, and I think it perfectly challenges a lot of the textbook chemistry we just covered today.
You're talking about the storage lesion debate.
Yes, the storage lesion debate in blood banking.
It's a fascinating paradox. This raises an important question, really. When donated blood sits in cold storage at a blood bank, its chemical composition begins to degrade over time.
Naturally, yeah.
And one of the most rapid changes is the depletion of 2,4,3-BPG, that same exact molecule we mentioned earlier that helps adult hemoglobin drop its oxygen into the tissues.
Right, because 2,4,3-BPG pushes the hemoglobin into the tense state, the T state, so it lets go. So without it, the hemoglobin is trapped in a higher affinity relaxed state.
Precisely. So based on the fundamental chemistry we just outlined, hemoglobin without 2,4,3-BPG holds on to oxygen way too tightly.
Okay.
Therefore, older banked blood should be functionally useless. I mean, it might carry the oxygen, but it won't release it effectively to a critically ill patient who desperately needs it.
The textbook math says it should be a total failure.
Right. But that's not what happened in the real world.
Wait, really? What did they find?
In the 2010s, researchers conducted these massive randomized controlled trials comparing fresh blood to older 2,4,3-BPG depleted blood in critically ill patients. And the results shocked the physiological community. There was absolutely no clear clinical harm.
Wow.
The patients receiving the older, theoretically broken blood survived at the exact same rates.
So the models predicted failure, but the human body somehow absorbed the chemical deficit and dynamically compensated for it.
Exactly. The body found a way around the textbook limitations, whether it was through shifting blood flow, increasing extraction ratios, or maybe some other allosteric backup system we haven't even fully documented yet.
That's incredible.
The rigid biochemical rules were essentially bypassed by the sheer adaptability of human physiology.
And honestly, that is a perfect thought to leave you with today. I mean, we have mapped the hemoglobin molecule down to the literal atomic level.
On account of the salt bridges.
We know exactly how it shifts, how it binds, how it collaborates with enzymes moving a million times a second. But even with all of that microscopic knowledge, the body still holds on to its secrets.
It certainly does.
Because if the fundamental chemical models say this stored blood shouldn't work well, but the human body somehow seamlessly rewires its biology and survives anyway, what other invisible backup systems, metabolic workarounds, and biological safety nets is your body running right this very second that the textbooks haven't even figured out yet? You are so much more resilient than the math suggests.
Biology always reserves the right to surprise us.
It really does. Well, until next time, keep diving deep.